Hello students, Mrs. Kleins here with your next section of video notes over the cell membrane and membrane transport. Just a couple quick reminders before we get started. Remember that you can pause this video at any time. You can also rewind this video to recap any concepts that you may have missed the first time. Let's get started with the cell membrane. So what is the cell membrane? We say that the cell membrane is selectively permeable, meaning that it regulates what enters and leaves the cell. We can see in the image on your screen that it can also act as a protective barrier, keeping the internal environment of the cell separate from the external environment. And because of that, it helps the cell to maintain homeostasis. Remember that homeostasis means the cell can maintain a steady or stable internal environment by regulating what can enter and leave the cell. The cell membrane goes by many names. It is sometimes called the plasma membrane, the selectively permeable membrane, or sometimes the phospholipid bilayer. A membrane can also be found around different organelles within the cell, such as the nucleus, mitochondria, chloroplast, and lysosomes. So what is the composition of the cell membrane, or what is it made of? As I stated earlier, one of the names we call the cell membrane is the phospholipid bilayer. The reason we call it that is because it is made of two layers of lipids called phospholipids. A phospholipid contains one phosphate group and two fatty acid tails. The phosphate head is hydrophilic, which means it loves water or it is happy coming into contact with water. The tails are hydrophobic and nonpolar, which means that they fear water or are repelled by it. So in the structure of the phospholipid bilayer, the fatty acid tails are attracted to each other, meaning they face each other. This gives the hydrophobic portion of the membrane on the internal part of the phospholipid bilayer. The superficial part that face the outside or inside of the cell are phosphate heads, which are hydrophilic or water-loving and are polar. So the important feature of the phospholipid bilayer is the membrane prevents things from being able to easily diffuse through unless it has some very special properties. Here is another image showing the phospholipid bilayer, and again, this would be like the outside of the cell, where the phosphate heads are, and this would be the fatty acid tails that are facing each other, almost like a sandwich structure. The cell membrane is fluid and dynamic in nature, meaning that the components of the cell membrane can kind of move around. So it's got a lot of flexibility, as you can see here with this image of a phospholipid bilayer. The phospholipids don't split or fall apart, but it also has this mosaic property, meaning that there's other components embedded within the phospholipids, and these little structures can float around almost like rafts on water. The different components of the cell membrane can include proteins. These proteins can aid in transport, like moving materials across the cell membrane, or they can just aid in the membrane structure. In the cell membrane, you'll have integral proteins, like this one seen here, which go all the way through the membrane. You also have peripheral proteins that you'll find on one side of the membrane or the other. The last type you could see are channel proteins that almost have a little tube or a pore that run through them and allow substances to pass through. There are also carbohydrates, which are very important for cell identification and recognition. You can see some of these glycoproteins, which are a protein with a carbohydrate tail sticking off. These structures can be used for identification. A good example of that is your immune system. It is how your immune cells can identify bacteria cells or viruses or structures that are foreign to your body and create an immune response to keep you healthy. Another would be a glycolipid. Glycolipids are going to be lipid structures with a carbohydrate attached to them. Another feature of the cell membrane that we will talk about is also a lipid and is known as cholesterol. Cholesterol in the cell membrane helps to keep it from becoming solid. It keeps its fluid feature and dynamics and keeps the cell from sticking to itself. You can see the cholesterol here. Seeing these structures again in other images, you can see your channel proteins, as seen here. You can see your peripheral proteins, seen on one side or the other. or your integral proteins that go all the way through. You can see glycolipids, like this one here, with a carbohydrate attached straight to the lipid bilayer, or glycoproteins, with a protein and a carbohydrate tail attached to it. Remember, these carbohydrate chains are used for identification and for signaling in the cell. Now that we're more familiar with what the cell membrane is, we'll talk about how it transports materials. So, before we get started, it's important to understand the concept of a concentration gradient. A concentration gradient occurs when the concentration of particles is higher in one area than in another. We can see in this image, when a substance is said to go with the concentration gradient, it means it's going from an area of high concentration to an area of low concentration. This does not require energy, and this would be similar to you spraying perfume in a room. Where the perfume is sprayed would be the high concentration area, and then it diffuses out across the room to areas of lower concentration. The other observation we can see with the concentration gradient is when things go against the concentration gradient. This means they're going from an area of low concentration to an area of high concentration. And as you can see in the image, this is going to require energy. We'll talk about how this can occur within our cells and across the cell membrane. The first type of membrane transport we're going to discuss is passive transport. Passive transport is going to move the molecules with the concentration gradient, meaning that we're moving molecules from an area of high concentration to an area of low concentration. This is passive transport because it does not require an energy source. So you can see here, we've got the membrane represented in pink. To start out, we have the high concentration of particles on the left, low on the right, and over time, they diffuse until they reach this equilibrium, where it's the same concentration on both sides of the membrane. There are three types of passive transport to be familiar with: diffusion, osmosis, and facilitated diffusion. Diffusion or simple diffusion is just known as the movement of particles from an area of high concentration to low concentration directly across the membrane. No assistance is needed in transporting these molecules. An example would be when oxygen molecules diffuse across the membrane to enter the cell so the cell can use the oxygen, or when carbon dioxide diffuses across the membrane to leave the cell. Osmosis is specifically the movement of water across the cell membrane. So as we can see here, these little molecules represent water, or H2O, and these molecules represent solutes, or substances that are dissolved in the water solution. Here we see that the water is going to move with the concentration gradient of water molecules from where there's more water to where there's less. Generally, what we observe is that water is going to move towards the area where there's higher concentrations of solute, or substances that are dissolved within the water. Facilitated diffusion is the third type, and it's similar to diffusion, but it requires a membrane transport molecule like a channel protein, as we saw in your earlier slide. These membrane transport proteins help move sometimes larger molecules across the cell membrane, such as glucose, or sometimes they open and close at certain times to allow substances to cross from areas of high concentration to areas of low concentration as needed. A good example of osmosis happening in our bodies in real time would be like when gums get swollen or irritated, and that's because we get extra water in the tissue of our gums. You can see here there's a high concentration of water in the gums and a lower concentration of water inside the mouth. These red molecules represent salt. So when you rinse with salt water, that water concentration is lower, and water is going to move out of the gums towards the area where there is a higher concentration of salt or lower concentration of water. That allows the gums to lose water but also become less inflamed or swollen. That would continue to happen until you have the same concentration of water on both sides of the membrane, meaning you have reached equilibrium. Active transport is the movement of molecules against their concentration gradient, meaning it is molecules moving from a low to high concentration, and because things are moving against the gradient, it's going to require energy. When we talk about these events happening in the cell, that form of energy is going to be ATP. In the future, we'll talk about how ATP is made or synthesized. So the three types of active transport to be familiar with would be sodium-potassium pumps, endocytosis, and exocytosis. We are going to start with sodium-potassium pumps. You can see here in this first figure that we've got this pump that opens and closes on both sides of the membrane. But what the pump does is it grabs up sodium in the area of low concentration and transports it to where there's a higher concentration of sodium. And at the same time, when the pump opens on the other side, it's going to transport potassium across the cell membrane. And what that does is create this separation. And when that happens, it generates an electrical potential, similar to what we would see in a battery. This is going to be like how our neurons are able to communicate with each other and send electrical impulses by creating that separation of molecules or charges. Endocytosis is going to be the mechanism by which large molecules move into the cell. So you can see these purple molecules are large, and when endocytosis occurs, you see this vesicle-like structure that forms around the substance, and that's part of the cell membrane that forms like a pocket and then kind of forms a bubble, where the bubble pinches off to form a structure called the vesicle. This will allow these substances to be transported deeper within the cell, perhaps to a region where they're needed for some important cellular function. The third type of cell membrane transport is exocytosis. This is the opposite of endocytosis. We've got these vesicles that contain substances, usually materials that the cells make and package, like proteins. But those vesicles travel to the surface of the cell membrane where they fuse with it and they release the material outside of the cell. So the membrane that was once part of the vesicle now becomes part of the cell membrane. So just kind of pulling this all together, membrane transport gives cells the ability to maintain homeostasis by allowing them to bring materials into the cell and release materials out of the cell. These functions are necessary for the cell to maintain homeostasis and perform its role in the human body. Cells are specialized, so there's different types of cells, but that also means that they can perform membrane transport in various ways. So I explained earlier that the nerve cells or neurons in our brain have lots of these sodium-potassium pumps in order to send impulses by creating that electrical potential. Blood cells in your body, on the other hand, are really important for transporting oxygen. So oxygen easily diffuses across the blood cell membrane into the cell so it can be transported. The oxygen can be transported from the lungs inside the red blood cells to the other tissues in the body that require it, and that occurs through simple diffusion. Okay, that wraps up this section of notes. As always, if you have any questions, just make sure to ask them when I see you in class.